Physicochemical principles of human cells in malignant transformation. We have extensive knowledge about the composition of cells, from the largest molecules to the tiniest ones. Advanced machines are capable of reading genes from a single cell. It seems that we are close to fully understanding life through the lens of biological science. What is life, and how is it created? The answer may seem straightforward: our genes are the blueprints of life. However, life is not solely created by genes. Genetic factors do not cause most diseases. Over 60% of birth abnormalities do not have specific gen links. Only 3% of individuals diagnosed with breast cancer have a mutation in the BRCA1 or BRCA2 gene. DNA does not explain why the heart is on the left side, how many fingers we should have, or how our cells divide when genes are completely locked inside chromosomes. Dr. Arias highlights that we are composed of an intricately complex and constantly changing symphony of cells. This raises the following question: what constitutes a cell? It is well known that proteins form cells. Carbohydrates, fats, proteins, and nucleic acids are produced and operate with the assistance of proteins. The function of genes depends also on proteins. The image shows how 5 different enzymes are involved in DNA strand duplication. Many proteins still have unknown functions, and their specific role is a mystery. Our understanding of how proteins interact with one another is still quite limited. The cooperative functioning of proteins and other compounds is incredibly complex. The chemical processes they perform vary widely. As a result, the principles of physics and chemistry often seem not to apply within the cellular environment. A cell contains millions of large and small molecules packed into a volume smaller than a drop of water. This raises a crucial question about how a cell's components work together to sustain life. In this video, we will explore this question by examining the known laws of physics and chemistry. Through this exploration, we aim to uncover chemistry, what the malignant transformation of a cell is, and how cancer cell develops. In his 1944 book What is Life?, the renowned physicist Erwin Schrödinger wrote, "The functioning of the body relies on precisely defined physical laws. These physical and chemical laws are grounded in atomic statistics, and their accuracy stems from the large number of atoms involved. The arrangement of atoms in the body and their interactions fundamentally differ from those of the objects that physicists and chemists have studied until now. However, this fundamental difference might seem unimportant to anyone who is not a physicist. A person without a background in physics is unlikely to recognize variations in statistical structure when expressed in abstract terms. It's challenging to comprehend how a single cell, composed of countless substances, can facilitate numerous chemical reactions at once, forming millions of new substances every second. This intricate process has contributed to our physical health for many years. All these activities originate from a single cell at the very beginning of the embryo. Regardless of terminology, all cellular functions ultimately stem from chemical reactions. In short, the life of a cell is the sum of chemical reactions and consists of synthesis and degradation. The synthesis and breakdown of substances happen at a specific rate, known as the rate of chemical reactions. In the human body and within cells, temperature and pressure remain constant. Therefore, all reactions must occur with the involvement of catalysts. Catalysts are protein compounds known as enzymes. The chemical reactions that occur within a cell are referred to as biochemical reactions. This term highlights reactions that occur at normal temperature and pressure, assisted by enzymes, which differs from chemical reactions in a laboratory flask. The chemical reactions within a cell depend on the availability of nutrients, which consist of various types of chemical compounds. This series of chemical reactions is referred to as metabolism. Metabolism is divided into 2 main categories: anabolism, which involves synthetic processes, and catabolism, which focuses on the breakdown of substances. Anabolism and catabolism refer to the chemical processes of synthesis and breakdown within the body. These terms describe the various chemical activities that occur in cells. To illustrate this, we can compare a cell to a chemical production facility. For example, consider it a complex dedicated to producing many metals. Plant management or administration is at the center of the operation, determining what to produce and in what quantities. This administrative function is separate from direct production. Surrounding this core are production laboratories and workshops that extract various metals from complex ore, including gold, silver, lead, and others. Based on demand, external factors determine what to produce and in what quantities. To manufacture products, a plant requires raw materials and financial resources, specifically investments. Similarly, a cell operates in the same manner. The cell also produces products, such as proteins and other essential components. The nucleus is at the center of the cell, much like the administration of a factory. The nucleus is separated from the cytoplasm, where production takes place. The nucleus contains the genetic material that determines what and how much the cell should produce. The demand for specific types and quantities of products comes from outside the cell, according to the body's needs. The cytoplasm contains various organelles, including the endoplasmic reticulum, Golgi apparatus, mitochondria, and lysosomes. It is rich in ribosomes and polysomes. Organelles and protein complexes have specific functions and produce essential products such as proteins, fats, carbohydrates, many other compounds, and numerous combinations of these chemical substances. Energy is required to produce products or synthesize chemicals. The cell's primary energy source is adenosine triphosphate. Now, let's explore how energy is produced in the cytoplasm. The fastest energy production method is glucose breakdown, known as glycolysis. Glycolysis takes place in the cytoplasm with the assistance of specific glycolytic enzymes. During glycolysis, glucose is converted into pyruvic acid, producing 2 molecules of ADP. Pyruvate produced during glycolysis can enter the mitochondria and break into water and carbon dioxide. Mitochondria have a double membrane structure. The outer mitochondrial membrane, which encloses the entire organelle, contains large numbers of membrane proteins and enzymes involved in diverse activities. The inner mitochondrial membrane contains proteins that perform the electron transport chain redox reactions, ADP synthases, and transport proteins that regulate metabolite passage into and out of the mitochondrial matrix. The matrix is the space enclosed by the inner membrane. It is important in ADP production with the aid of the ADP synthase. The matrix contains hundreds of enzymes. The enzyme's major functions include the oxidation of pyruvate and fatty acids and the Krebs cycle. In the mitochondria, fats and sugars are oxidized using oxygen, resulting in carbon dioxide and the formation of a high-energy phosphorylated compound ADP. This process is referred to as oxidative phosphorylation or respiration. When we breathe, we provide mitochondria with oxygen, which they use to generate energy. As a result, we exhale carbon dioxide. The energy needed to convert ADP into ADP is derived from the proton gradient across the mitochondrial membrane, facilitated by the electron transport chain. This process creates the energy necessary for synthesizing organic compounds essential for survival. Cell formation occurs through chemical processes in the cytoplasm, utilizing the energy supplied by ADP. Therefore, understanding the cytoplasm is essential in conjunction with studying genes. For the past 5 decades, researchers have primarily studied genes to find a cure for cancer. However, genes without the chemical processes occurring in the cytoplasm are like a library without any readers. If we think of the genome as a musical score, the orchestra and the performers are in the cytoplasm. However, this orchestration remains largely mysterious and operates like a black box. Chemical reactions in the cytoplasm lead to the formation of various complex life forms, including crustaceans. However, malignant cell types, such as cancer, can also develop. What is the difference between the normal chemical processes that create life and the abnormal chemical processes that occur in cancer cells? Can cancer develop due to disruptions in the chemical processes within the cell's cytoplasm? What is the role of processes that create energy, including glycolysis and oxidative phosphorylation, as a direct supplier of ADP for chemical reactions? Cancer is often called the emperor of all maladies because it arises from cellular pathologies. It is a disease that originates from cell dysfunction. Cancer is defined as a disease or pathology of the cell, which indicates that it violates normal chemical reactions. In short, cancer can be described as a disease characterized by uncontrolled cell division due to violating normal chemical reactions. Gene mutations that transform normal cells into cancerous ones can explain the cause of cancer. This transformation signifies a disruption of genetic instructions, leading to abnormal chemical processes. If cancer is characterized by unregulated cell division, we should consider the underlying question: why do cells divide? Is the mutation triggering this malignant division, or could other factors be at play? Cells divide according to well-understood patterns. The phase when a cell is non-dividing is known as the G0 phase of the cell cycle. G0 is called the resting phase, during which the cell is neither dividing nor preparing to divide. For a cell to enter division, it must move into the active phases of the cell cycle. The cell cycle is the series of events that a cell undergoes every time it divides, creating a new daughter cell. This cycle consists of several distinct phases, each fulfilling a specific function. The longest phase of the cell cycle is interphase, which includes the G1, S, and G2 phases. During interphase, the cell prepares for mitosis. The key phase of interphase is the G1 phase, during which the cell actively synthesizes the mRNA and proteins needed for mitosis. In this phase, the cell grows and prepares itself, determining whether it will begin dividing. The S phase is regarded as the most critical phase. Once the cell enters the S phase, the progression becomes more technical. Synthetic processes, particularly protein synthesis and cell volume growth, are essential for normal and cancer cell division. Consequently, the rate of chemical processes, especially protein synthesis, is crucial in determining cell division. Consider this analogy involving a polymetallic plant. If there is a substantial increase in demand for the plant's products and it begins to produce more, eventually, the plant will establish a subsidiary by creating a new facility. To better understand how a cell functions, we can compare it to a polymetallic plant. When a cell produces an excessive amount of proteins and other substances, it can become overcrowded. The only solution in this case is for the cell to divide. The overproduction of proteins is prompted by the rate of chemical reactions occurring within the cell. In biological terms, it is determined by the speed of metabolic reactions. The next logical question is, what factors influence the rate of chemical reactions in a cell? Since all cells in the body receive the same amount of nutrients and signals, why do cancer cells divide more frequently and operate without regulation? First, let's examine how we understand biochemical reactions. Since all biochemical reactions are enzymatic, biochemists focus on calculating their rates. Canadian scientists Michaelis and Menton were the first to develop a method for calculating the kinetics of enzymatic reactions. The Michaelis-Menten constant is crucial for determining reaction kinetics. For practical purposes, the Lineweaver-Burke plot is often used to transform the Michaelis-Menten equation to facilitate analysis. This plot allows for straightforward calculations of the rate of chemical reactions involving a purified enzyme in an aqueous solution. Additionally, the Lineweaver-Burke plot is important for assessing enzyme purity. It is also helpful in drug development, as it helps to evaluate the effectiveness of enzyme inhibitors. These graphs and equations do not function as expected in a cell. Why is that? To understand this, we must first consider the cell's structure and the cytoplasm's physicochemical state. There are several reasons why the well-known laws of physics and chemistry do not work in cytoplasm. Chemical processes in the cytoplasm occur within a dense environment. The cytoplasm is an extremely heterogeneous and viscous medium. Viscosity is a measure of a fluid's resistance to flow. A fluid with high viscosity is thick and resists movement. Examples include ketchup and honey. The cytoplasm is classified as a non-Newtonian fluid, which means its viscosity changes when force is applied. For example, ketchup becomes more fluid when shaken, illustrating the properties of a non-Newtonian fluid. Furthermore, the cytoplasm is a viscoelastic fluid, displaying both viscous and elastic characteristics when it is deformed. A viscoelastic fluid possesses properties of both liquids and solids, exhibiting viscosity and elasticity. Honey is an excellent example of a viscoelastic fluid. In a viscous medium, diffusion is restricted, limiting the free movement of molecules. Diffusion is the process by which molecules move from areas of high concentration to low concentration along a gradient. When free diffusion is restricted, it can slow down the rate of chemical reactions. The main barrier to the free movement of molecules is the medium's viscosity. The Stokes-Einstein equation describes the relationship between viscosity and diffusion. It suggests that the thermodynamic force driving a particle's movement is balanced by the resistance of the fluid. According to this equation, the diffusion coefficient is inversely proportional to the viscosity of the liquid, assuming constant temperature and molecular radius. Fluctuations of small particles, random oscillations, in a liquid medium are called Brownian motion. The Stokes-Einstein theory explains the Brownian motion of particles in a liquid medium. While a cell can be considered a fluid medium, the movement of molecules in the cytoplasm does not adhere to the Stokes-Einstein principles due to the varying sizes of the molecules. Although there is a high concentration of various substances in the cytoplasm, the diversity of these substances limits the number of molecules that react. As a result, one of the fundamental principles of chemistry, known as the law of mass action, may not hold. The law of mass action states that the rate of a chemical reaction is proportional to the active masses of the reactants, provided the temperature remains constant. Active masses refer to the substances that actively participate in a chemical reaction. If free diffusion is limited and the number of actively interacting molecules is minimal, how can millions of chemical reactions occur in a cell every second? Does this contradict the laws of chemistry? Understanding how chemical processes can happen within a cell without violating physico-chemical principles is crucial. To address these inquiries, scientists have conducted careful studies of cell structure, particularly focusing on the cytoplasm, over the last century. The introduction of electron microscopy marked a significant advancement in structural analysis. While EM is a powerful tool, it does have limitations. One major drawback is that it requires fixed or dead cells, which can introduce artifacts during the fixation and staining processes. Additionally, the heavy metals used in electron microscopy can bind to lipids, potentially affecting the results. All organelles contain lipids, creating the impression that the entire cell primarily comprises lipids. This is evident in electron micrographs of cells, where we mostly see the outlines of lipids rather than the actual structures of the cell. Proteins that are not detectable by electron microscopy play a crucial role in maintaining cellular structure. An electron microscope image was obtained from a cell after removing lipids using a detergent. The detergent dissolved the endoplasmic reticulum, the outer membrane of the nucleus, mitochondria, and all other lipid-based structures. Then, the cell interior revealed detergent-resistant filaments associated with ribosomes and mRNA. Dr. Porter used a high-voltage electron microscope to investigate cell structure. He proposed a cytoplasmic structure called the microtrabecular lattice, which he observed using this advanced imaging technique. Due to the histological techniques, many researchers viewed the new cytoplasmic image as an artifact. In the 1970s, the concept of the cytoplasm was introduced as a macromolecular network, matrix, or lattice arranged in a solid phase rather than being viewed as a solution or cytosol. Unfortunately, this idea has not been confirmed through experimental means, as the lattice or matrix has not been chemically isolated. Even if the matrix could be isolated, determining its content and composition would still be impossible. Dr. Schlieve stated, Heisenberg's uncertainty principle in quantum mechanics applies to cytoplasmic structure as well. Will we ever truly see the cytoplasm's structure? Perhaps not, but we can certainly get closer. After 20 years of persistent research, it has been concluded that further studies must be postponed until new technologies are developed to identify the types of proteins and their associated components. The statement highlighted the importance in explaining cellular processes through the lens of established laws of physics and chemistry. In summarizing the scientific approaches from previous centuries and contemporary concepts, it is essential to emphasize the importance of the physical and chemical state of the cytoplasm in understanding the structure of the cytoplasm. Dr. Pollack points out that textbooks often start by describing the cytoplasm as a gel, but then they discuss chemical mechanisms based on the idea of free diffusion, treating it as if the cytoplasm is simply an aqueous solution. This inconsistency occurs because the behavior of gels is not well understood, particularly among biologists. Recent computer models indicate that the cytoplasm exhibits sponge-like poroelasticity, Although the cytoplasm is significant, our understanding of it remains limited. To determine whether the cytoplasm contains a solid phase, separating the solid components from the liquid ones is essential. What methods can we use to achieve this separation? One important concept to consider is the impact of inorganic salts on proteins and macromolecules in aqueous solutions, explained by the Hofmeister series, discovered in 1888. The Hofmeister series classifies ions based on their ability to either dissolve or precipitate proteins, depending on the concentration of each specific ion. In the cell, potassium serves as the primary positive cation. These ion concentrations were used to separate the cytoplasm into distinct phases. This phase separation demonstrated that the cell comprises both an elastic solid phase and a viscous liquid phase. Chemical reactions within a cell differ significantly from those occurring in flasks. The rate of these cellular reactions is influenced by extracellular signals, which act through the mechanical processes of the cytomatrix. The mechanical movement of the cytomatrix helps to mix the contents of the cytosol, ensuring that nutrients and substrates are distributed throughout the cell. In this way, the cell overcomes the viscosity of the medium and the scarcity of the number of molecules. Thus, physicochemical laws are not violated. Mechanical mixing is replaced by mixing with motor proteins. In this case, the cytoplasm, as a viscoelastic, non-Newtonian fluid, obeys the laws of physics, becoming more fluid under the pressure of motor proteins. However, the incoherent effect of all active processes also creates randomly fluctuating forces, which drive diffusive-like, non-thermal motion. Using force spectrum microscopy, shows that force fluctuations substantially enhance intracellular movement of small and large components. The evidence is that blebbistatin treatment and ADP depletion suspend the movement of particles. ADP depletion or treatment of a myosin inhibitor would not affect the motion of the particles in Brownian motion. The fluctuations of the cytoplasm are not Brownian thermal motions. The fluctuations are 3 times larger in malignant cells than in benign counterparts. The active force is used to move molecules in the cell. What is the difference between cytosol and cytomatrix? Mass spectrometry analysis of protein composition revealed differences between the cytomatrix and cytosol protein spectra. Cytosol and cytomatrix proteins were categorized based on their functional properties. Approximately 7,000 proteins were analyzed. The proteins that serve as the structural basis of the cytomatrix, including actomyosin and other actin-binding proteins, are the most numerous and diverse among structural proteins. They account for approximately a quarter of all structural proteins identified. In non-muscle cells, actin constitutes 1% to 5% of total cellular proteins. In muscle cells, this percentage can increase to as much as 10%. The isoforms of actin, namely beta-actin and gamma-actin, are predominantly found in non-muscle cells. The actin cytoskeleton comprises 6 types of actin, over 40 types of tropomyosins, 15 classes of myosins, and various other actin-binding proteins. These actin-binding proteins play a crucial role in regulating the rate of actin polymerization and determining the length of actin filaments. Monomeric globular actin is an ATPase with a mass of 43 kDa. G-actin can self-assemble into filamentous actin. ADP hydrolysis is necessary for both the assembly and disassembly of F-actin. Additionally, ADP hydrolysis regulates the interaction with various proteins. Approximately 150 proteins are known to regulate the activity of actin. Because of the self-assembling capacity of actin into polymeric fibers with ADP, many actin regulators primarily inhibit this self-assembly. The supposed active strength of the cytomatrix is actin and myosin. In this way, the active force of motor proteins overcomes the diffusion limitation of the viscous medium. The law of the acting masses is not violated, either. Numerous post-translational modifications of proteins by lipids, carbohydrates, and polysaccharides serve to stabilize the cytomatrix in a solid phase. Actin, in particular, is remarkable for having the highest number of these modifications, with 17 different types. This raises the question, why does a filamentous fibrillar protein require so many additional modifications that could potentially disrupt its geometric structure? Post-translational modification of actin plays a crucial role in its ability to generate active force. Figuratively speaking, actin can be compared to a rod with blades. It can stir a viscous liquid, transforming it into a flowing state. For the intensive interactions between actin and myosin, such as those occurring in muscles, energy in the form of ADP is required. Assembly of another motor protein, tubulin, requires GTP rather than ADP. This difference in nucleotide preference could be crucial in determining which motor protein is selected for mechanical activities within the cytomatrix. The next most prevalent categories of proteins include transmembrane proteins and those associated with the endoplasmic reticulum and Golgi apparatus. Consequently, membrane-forming structures such as the Golgi apparatus, and plasma membrane are integral components of the cytomatrix. The cytomatrix is rich in various growth factor receptors and tyrosine kinase receptors, indicating a connection between the intracellular and extracellular matrices. Signals of calcium, cyclic adenosine monophosphate, and G proteins are plentiful. Various transport pathways traverse the cytomatrix. This list of transport systems supports the concept that the intracellular cytomatrix consists of structures facilitating both intracellular and extracellular transport of molecules. Oncogenic signals can be transmitted through proto-oncoproteins and tyrosine kinase receptors. These signals are organized as protein complexes associated with the cytomatrix. The family of oncogenes, including RAS, SRC, MYC, BRCA, CBL, JUN, DEC, and other tumor-associated proteins, is among the most numerous and diverse. The presence of proto-oncogenic proteins in the cytomatrix highlights their critical role in the formation of malignant cells. The diverse range of enzymes in the cytomatrix suggests that metabolic reactions, which could potentially inhibit each other, may occur simultaneously due to their immobilization on the cytomatrix. Immobilization and segregation can help overcome spatial barriers to chemical reactions. Immobilized enzymes are very effective and widely used in industry. Immobilized enzymes require a flow of substrate solution. Enzymes attached to the cytomatrix require the flow of cytosol for their reactions. The actomyosin dynamics can lead to highly directional movements, creating a tunneling effect. This tunneling effect addresses the limitations posed by a gel-like structure and the crowding of molecules. Ribosome profiling or ribosome imprint or RiboSeq. The method known as ribosome profiling, or RiboSeq, is a valuable technique for analyzing the functions of cytomatrix. We compared the active and inhibited pathways in the cytomatrix with those in the cytosol. The localization of calcium and phosphatidylinositol signaling pathways indicates that the endoplasmic reticulum and plasma membrane are integral components of the cytomatrix. Different signaling pathways and non-receptor signaling pathways, focal adhesion, gap, and contact junctions are active in the cytomatrix but suppressed in the cytosol, which can influence the dynamics of actomyosin. The study of inhibited cytomatrix pathways revealed a distinct range of gene sets activated in the cytosol. These pathways include tRNA biosynthesis, the cell cycle, carbohydrate metabolism, and the degradation of amino acids and proteins. Additionally, glycolysis, gluconeogenesis, RNA degradation, nucleotide metabolism, and DNA repair and splicing pathways were enhanced in the cytosol but reduced in the cytomatrix. For the DNA repair and splicing pathways to be transferred to the nucleus, they must not be associated with the cytomatrix. Data from mass spectrometry and ribosomal profiling indicate that cells contain two phases, liquid and solid. Both phases are dynamic. The solid phase functions similarly to a pump, like how the heart circulates blood throughout the body. This invisible movement requires a significant amount of energy. The force needed to move the viscous cytosol must be substantial, as it facilitates over 1 million chemical reactions per second within the cell. Consequently, the speed of movement of the cytomatrix and cytosol can impact the rate of chemical processes and metabolism. This metabolic rate ultimately influences whether cells grow normally or develop cancerous characteristics. The matrix system of biocatalysis plays a crucial role in facilitating efficient responses, transport, and signaling in healthy tissues under normal physiological conditions, primarily through mitochondrial respiration. In contrast, cancer cells, due to their limited number of mitochondria, predominantly rely on the anaerobic pathway for energy production glycolysis. As a result, malignant cells form lactic acid lactate. Why do cancer cells exhibit increased glycolysis? Typically, the breakdown of glucose into pyruvate and lactic acid production occurs in leg muscles during intense running. In these instances, pyruvate is converted into lactic acid. Muscle cells rely on glycolysis to meet their heightened energy demands. Lactate is a byproduct of this process. Lactate can then be converted back into glucose through a mechanism known as the Cori cycle. Tumor cells are not directly involved in the body's physical activity but produce lactate, similar to muscle cells. The energy value of the Warburg effect was previously underappreciated because of a lack of understanding of its mechanical processes. It was unclear where the excess energy from glycolysis was utilized. With the discovery of the cytomatrix, an elastic solid phase of the cytoplasm, we now know where this excess energy is directed. Similar to muscle cells, malignant cells share the energy needs primarily to drive the provision of ADP to actin. While there are notable differences in the content and regulation of non-muscle fibers compared to muscle, the actin-myosin interactions in non-muscle fibers may also contribute to lactate production. Consequently, the glycolytic pathway is crucial in adapting to changing energy demands. Cancer cells are characterized by the overproduction of proteins and increased ribosome biogenesis, manifesting as nuclear hypertrophy. These cells require more energy than a typical number of mitochondria in a malignant cell can supply. To meet this heightened energy demand, cancer cells utilize glycolysis, the new concept of cellular chemistry and cancer. Mutations and epigenetic changes can lead to imbalances in protein levels within cells, promoting tumor heterogeneity. Cancer cells increase protein synthesis to compensate for lost functions. The heightened activity of the cytomatrix's micromechanics can stimulate biosynthetic processes, leading cancer cells to demand more energy production. If their energy needs are not met, cancer cannot develop, even in the presence of mutations. Additionally, if the activity of motor proteins, particularly actomyosin, is controlled, cancer is less likely to emerge. As the micromechanics of the cytomatrix plays a crucial role in regulating the rate of chemical reactions. The necessity for changes in matrix mechanics to fulfill the biosynthetic and energy requirements of malignant cells indicates crucial transformations in cancer development. Thus, alterations in matrix mechanics and oncogenic mutations are essential for the onset and progression of cancer. The activity of motor proteins in the cytomatrix must be significantly higher than that of normal cells. Therefore, it is not only oncogenic mutations that are necessary for cancer progression. Clinical cancer is rare, even though gene mutations occur frequently. Most mutations that do not lead to cancer are classified as passenger mutations. The reason some mutations result in cancer while others do not remains unclear. So far, more than 500 genes have been identified as potentially able to cause cancer when mutated, but these genes do not all have the same cancer-causing potential. This uncertainty can be attributed, in part, to chance, while the physical and chemical processes within the cell are often overlooked. A two-phase system of the cell's physical state may help clarify this uncertainty. Cells' ability to extract additional energy is rooted in their chemical processes, with anaerobic glycolysis being one of the oldest ways of energy production. Cancer primarily arises from mutations that enable the cell to achieve a high state of energy production necessary for matrix mechanics. Cancer cells metaphorically acquire a musculature that demands more energy regarding cytomatrix mechanics. If mutations do not lead to increased energy output, cancer will not survive. The recurrence of cancer and the survival of cancer cells suggests that malignant cells have developed the ability to synchronize their energy production with matrix mechanics, allowing them to generate enough energy for tumor growth. This concept of tumor development highlights the role of the Warburg effect, genetic abnormalities, intracellular matrix mechanics, respiration, and glycolysis. Cancer recurrence happens when changes in the cytomatrix enhance its activity. Normal cell division can be explained using the following framework. Mitochondrial respiration and moderate matrix mechanics during normal biosynthesis lead to organized cell division. The characteristics of cancer cells can be outlined using the following framework. Mitochondrial respiration, anaerobic glycolysis, and enhanced matrix mechanics with accelerated biosynthesis lead to disordered cell division. An imbalance of protein synthesis due to mutation and an abundance of ADP due to glycolysis by enhancing the micromechanics of the cytomatrix can accelerate chemical processes, which can serve as a trigger for malignant transformation. The rate of chemical reactions in a cell is regulated, like the number of neutrons in a nuclear reactor, the disruption of which leads to unstoppable division. A new concept of cancer development elucidates unanswered phenomena associated with the disease. Why is the Warburg effect, also known as aerobic glycolysis, the primary energy source in cancer cells? Answer: the number of mitochondria in non-muscle cells is limited, making them unable to supply sufficient energy for malignant cells. Therefore, tumors rely on glycolysis. Would the same genetic mutations not cause cancer in everyone? Answer: the rate of biochemical processes depends on actomyosin activity, which controls chemical reactions necessary for normal biosynthesis. The activity of the cytomatrix remains regulated. Would those forming potential cancer cells not always trigger tumor development? Answer: the energy level is insufficient, or the activity of the cytomatrix is being regulated. Why is hereditary cancer fatal for some in childhood while others live to adulthood? Answer: the activity of the cytomatrix is regulated in hereditary mutated cells, as long as these cells remain normal. Would passenger mutations occur, and why don't oncogenes always trigger cancer development? Answer: the mutation rate is influenced by environmental conditions, time, and the cell's self-repair capabilities. Would cancer cachexia or weight loss by cancer patients happen? Answer: the energy derived from food consumption can gradually nourish cancer cells. Why is cancer fast growing in some cases but slow in others? Answer: the speed at which the cytomatrix engages in biochemical processes is a determining factor. The manifestations of cancer can be addressed by considering the following: increased mechanical fluctuations in the cytoplasm of malignant cells, strengthening chemical processes through mechanical processes, development and recurrence of cancer due to excessive energy acquisition by the cytomatrix, cancer resistance to chemotherapy due to physicochemical processes. The impact of drugs on cancer, insights from cytomatrix activity. Ribosome biogenesis and protein biosynthesis are two critical processes that determine the survival of cancer cells. The positioning of RPL7A on the 60's subunit surface makes it a valuable indicator for assessing the biogenesis of the large ribosomal subunit. We tested the effects of several compounds on ribosomal protein translation. To monitor the effects, we expressed GFP-tagged RPL7A in cells and observed the resulting fluorescence in living cells. Despite inhibiting various signaling pathways, these compounds all appeared to enhance RPL7A synthesis. In response to chemical agents, a significant quantity of newly synthesized RPL7A likely cannot penetrate the nucleus all at once, and some of it remains on the nuclear envelope. When suspended in the nuclear envelope, RPL7A forms a bright rim around the nucleus. As a result, the increased biogenesis of the 60's ribosomal subunit in the nucleus leads to nucleolar hypertrophy, which is caused by the excess of RPL7A. Automated tests indicated a significant increase in both the number and average size of nucleoli in the treated cells. The mTOR inhibitor, PP242, inhibits interdependent protein translation. Treatment of cell lines with PP242 caused many proteins to localize within the cytomatrix. Due to the observed changes in protein concentrations in the cytomatrix, it was important to determine whether proteins were moving from the cytosol to the cytomatrix or if protein synthesis in the cytomatrix was being enhanced. In this study, cells were treated with inhibitors of dynein and kinesin, and the levels of proteins in the cytosol and cytomatrix were compared. However, there was no significant exchange of proteins between the cytosol and cytomatrix. Polysome profiling is a technique used to assess the activity of ribosomes and polysomes. After treatment with the inhibitor, the polysome profiles in the cytosol and cytomatrix show opposite trends. In the cytomatrix, the levels of polysomes and large ribosomal subunits increase in response to the inhibitor. In contrast, in the cytosol, the levels of large ribosomal subunits and polysomes decrease. To examine the dynamics of the 40's small subunit under normal growth conditions and after treatment with PP242, the mRNA levels of the small ribosomal protein RP6 were measured using in situ hybridization. Even after 2 hours of PP242 treatment, cells showed higher levels of RP6 mRNA signals. To detect newly synthesized proteins, we employed the labeled amino acid method. Incorporation of L-phenylalanine into proteins demonstrated that the mTOR inhibitor PP242 reduced de novo protein synthesis in the cytosol by 20%. However, the incorporation of labeled amino acids resulted in a 50% increase in the synthesis of cytomatrix proteins. The results indicated an increase in the synthesis of new proteins within the cytomatrix, while protein translation was suppressed in the cytosol. Thus, using 6 different methods confirmed that protein biosynthesis can differ in the cytomatrix and the cytosol under the drug treatment. These findings suggest a mechanism for cell survival that involves shifting protein synthesis from the cytosol to the cytomatrix. This shift indicates that the coordination between cytosolic and cytomatrix functions is essential for the precise regulation of cellular processes. Notably, the inhibition of mTOR appears to promote protein synthesis in the cytomatrix, suggesting that protein synthesis remains stable even in the presence of drugs. Inhibition of mTORC1 triggers autophagy and proteolysis, the process of protein breakdown. Proteolysis occurs in the cytosol and provides amino acids that can stimulate protein synthesis in the cytomatrix. This reprogramming of protein translation from the cytosol to the cytomatrix is likely a vital mechanism for cell survival. Under extreme conditions, these functions can serve as a backup mechanism. Coordination of the metabolic processes between the cytosol and the cytomatrix may enhance cancer cell survival and contribute to drug resistance. Generally, the cytomatrix plays a key role in determining the cell phenotype and the type of tissue the cell belongs to. Using 6 biochemical methods and 3 advanced techniques, RNA sequencing, ribosequencing, and mass spectrometry, we demonstrated that when protein synthesis is restricted by a drug, protein translation is reprogrammed to occur in the cytomatrix. This reprogramming is a mechanism that aids in the survival of cancer cells. Suppressing the viability of malignant tumors by targeting the cytomatrix is crucial in effectively combating cancerous growths and can help prevent disease recurrence after treatment.
Thank you for watching this video about healthy and malignant cell chemistry. Without fundamental laws, the molecular processes in healthy cell may seem like a miracle, while pathological processes such as cancer can be compared to a cell that has gone mad, as described by a well-known oncologist recently. Understanding the nature of disease is essential for effective treatment. For instance, the development of antibiotics has been crucial in combating infectious diseases. As a scientist, I have dedicated myself to understanding the chemical nature of both healthy and malignant cells. For many years, I have purified enzymes related to human fat metabolism and isolated the protein complexes responsible for the survival of the malignant cells. This valuable experience has allowed me to develop a novel approach for dissecting the cytoplasm into 2 distinct components, a viscous fluid and an elastic solid phase. The elastic solid phase is a long sought component of cytoplasm that has been elusive for over 50 years, and I have named it the cytomatrix. Careful analysis of cytometrics using both conventional and advanced methods, including the examination of 7,000 proteins and 13,000 genes, has proved insights into how healthy cells function and how malignant cells develop and survive. Understanding cancer through the principles of physics and chemistry offers valuable insights into cellular functions. By examining the fundamental interactions at the molecular level, we gain a clear picture of how cancer cells emerge, grow, and spread. This approach paves the way for innovative treatments and therapies. Applying these scientific principles is crucial for advancing cancer research and improving patient outcomes. The treatment of cancer is highly challenging due to the difficulty of identifying the causes of malignant cell transformation. Although tumors share certain characteristics, such as uncontrolled division, nucleolar hypertrophy, and the Warburg effect, they are considered distinct diseases because of their different tissue origins and pathological forms. By linking the rates of chemical reactions in cells to the mechanical activity of the cytomatrix and the Warburg effect, we can gain a better understanding of the nature of cancer. This approach also allows us to explain cancer through the principles of physics and chemistry. Understanding the nature of cancer has allowed our team to develop new treatments and patent methods to combat malignant neoplasms. Targeting the cytometrics is crucial for effectively suppressing the viability of the malignant tumors and can help prevent disease recurrence after treatment. Our research has identified chemical compounds that significantly reduce the rate of chemical reactions within the cytomatrix. These compounds can serve as prototypes for future effective drugs. In conclusion, we are confident that we have identified the underlying reasons for cancer survival and resistance to therapy based on physicochemical processes occurring within the cell. By targeting the cytomatrix, we can inhibit the malignant growth and survival of cancer cells, as well as their resistance to drug treatments. Our results are published in publicly available scientific journals, and our methods are protected by patents. We have developed a biochemical algorithm to assess the effectiveness of anti-cancer drugs. This method enables accurate selection of drugs that can effectively prevent cancer recurrence. Pirinuk Labs is dedicated to developing drugs that specifically target the cytometrics. Join us in the fight against cancer.